Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. First of all, we'd like to take a moment and thank our Dr. GPCR Ecosystem partners for their support and help, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Tomorrow, we will be hosting another free symposium on GPCR activation and signaling. It's not too late for you to join us. All you have to do is become a Dr. GPCR site member, which is free. Our program includes talks from Oliver Hartley, Gaspar Pandey-Sekaresh, Ilana Kotliar, Nicolas Gilles, Shivani Sajdev, Etienne Billard, Surya Shinoy, Paul Gasser, Stuart Mosley, and Kavya Krishna Kumar. We also have set aside 2 hours for poster presentation and networking on a platform called Hopin for the symposium and for all upcoming symposium. If you haven't done so, there is a little bit of time for you to submit your poster and join us tomorrow. If you don't have a poster, that's okay. You can still come and network with your colleagues. For more information and an updated schedule, go to the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposium. You can join us live tomorrow starting at 8 a.m. To navigate the ecosystem, please also use the direct links in the footer. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm excited to have with me Fred Bruns. He is my guest in this episode. Fred, welcome.
Thank you. Thank you for having me on the podcast.
I'm very excited to talk to you today because you're an unusual guest, and I think throughout the episode, people are gonna figure out your career is not the traditional path of many of our guests, and I think that's what makes you having on the podcast very interesting. So, Fred, why don't you start by introducing yourself, please?
Sure. My full name is Robert Frederick Bruns Jr. I go by Fred, but I publish under Robert F. So just letting you know that. In terms of me, I have a PhD in neurosciences. I worked in industry most of my career. I retired in 2014, and I've been writing up some work that we did before I retired ever since. In terms of what the listeners might be interested in, I think that the most interesting things will be the bookends, what I would call the bookends of my career. In particular, at the very beginning of my career, my lab found the first positive allosteric modulator of a GPCR. Positive allosteric modulator is abbreviated PAM, so I'll say PAM a lot. And then at the end of my career, I was on a team that took a dopamine D1 receptor PAM into the clinic. So I think those are the really the highlights of my career, and I think what will be most of interest. In terms of college and education, I went to New College in Sarasota, Florida for a year. I started out as a physics major, and then I switched. I switched to Washington University in St. Louis, and also switched my major to psychology. Kind of a drastic switch, and I don't know, one of probably many unconventional decisions I've made during my career. So, for better or worse, that's what I did. I got an AB in psychology, and then went to University of California, San Diego for in neuroscience for my PhD. After that, I went to did a joint postdoc at NIH and Johns Hopkins. Then I went to work for Warner Lambert Park Davis in Ann Arbor, Michigan. It got swallowed up by Pfizer maybe 15 years after I went there, but I was already gone. I had left for Eli Lilly and Company at the time. So I worked for Eli Lilly until I retired at the end of 2014. So what else about me? I have kind of an Asperger type personality, for better or worse. Actually, a lot of I think scientists have something along that lines in terms of personality. I often get obsessed with ideas, And looking back, I think I often chose things to work on as though they were hobbies. So just, kind of looking back at things. Uh, as you might expect, I also have a hard time understanding people in social conventions. I've worked on it for 72 years, but I don't think I'm there yet. So that's just another thing about me. And as I mentioned, I've made what I think are some unconventional decisions throughout my life. And looking back, I kind of wonder how things might have turned out if I'd chosen differently. But I mean, that's obviously water over the dam at this point. In terms of grad school, how did I get interested in GPCRs? Well, I got interested in adenosine receptors after reading a paper in Pharmacological Reviews. It was called Purinergic Nerves by Jeffrey Bernstock. It was a very long paper, and it was all about all the evidence that ATP was actually a neurotransmitter. And starting from that, I soon found out that there was also an extensive literature on adenosine receptors. So I got interested in that. And for my PhD, I sent letters to chemists asking for adenosine analogs and also for analogs of caffeine. Caffeine was already known to block adenosine receptors at the time. So my dissertation was really the first large-scale structure activity study on adenosine receptors. And just as an aside, because it'll come up later, in 1977, I taught myself Fortran so I could write a program to analyze my data, so I could get the experiments done and finish up my PhD. So that's my story in terms of my education.
Thank you. And the reason I was mentioning that it's an unconventional career, because I don't think a lot of people can say that they were the first one to discover a PAM for a GPCR, and the one person who would take a PAM into the clinic. Also, I think that's your bookends are very, very unique and very interesting. Before we move on to the discussion, I wanted to stop a little bit and take us back to the time where you were mentioning that you started studying physics and then you switched to psychology. Mm-hmm. What got you interested in science in general, but why physics at the time?
Well, from the time I was very young, I can remember in second grade telling another boy that lived across the street from me that I wanted to be a scientist, and I kind of worshipped Einstein at the time. So that was my start there. So basically, as long as I can remember practically, I mean, I was in second grade at the time, I think. So as long as I can remember, I've wanted to be a scientist. And I've always had a good knack for math and for science and so forth. So that was how I first got into science. I was quite good at math. I actually got a perfect score on the math part of my SAT. So it seemed like I was headed for a career in physics, and then changed my mind for various, maybe idiosyncratic reasons. I kind of felt that neuroscience or psychology would be more interesting in some ways. The idea of how people think, how people communicate, all of that was of interest to me. So I made that switch. Looking back, I think one of the things maybe I might have thought more about at the time was picking a career that would build better on my math abilities, and maybe staying in you never know with the counterfactuals, but possibly staying in physics might have worked better, might have been a better match with my skills as well as my interests. So that's something to think about. I know you have a question later on advice for young scientists, and that's one of the things that I mentioned there.
But at the same time, you followed your passion. You followed what at the time you thought was the most interesting. And you made a comment when you were talking, saying that the projects you chose to work on were of a high interest to you at the time, and you didn't feel like you were working, it was a hobby. And I think it's every scientist's dream or every person's dream to be working on project they're very passionate about, because then you don't feel like it's actually work.
Yeah, that's definitely true. And throughout my career, that's been the case. Even when I was, kind of ended up in a job description I didn't really want, I still enjoyed the actual content of the job, regardless. So that's true. There are a lot of different things you can do that can be very fascinating in terms of things you can work on. And I haven't kind of got to it yet, but I was actually turned into a computational chemist at one point during my career, kind of the middle of my career. And so I've made a lot of changes over the course of my career.
But that shows also that you're very adaptable, and then you kept following the passion. And sometimes maybe you ended up in a role that didn't have the maybe a right job title, but the description and the activities that you had to perform in that role were something that challenged you and you actually enjoyed doing that.
Yes, that was always the case, regardless.
And
Yes.
that speaks to a lot of flexibility. And you mentioned advice to junior scientists, and I think one of those things is one other than follow your passion, is getting to know new things and learn new things. Would surprise you. Sometimes you might enjoy it.
Yes, definitely. And I also have advice for mid-career scientists, that studies have shown that most of the revolutionary discoveries in areas like physics are made by younger scientists, but there is one big exception, and that is when a mid-career scientist changes to a new field, and then they see things with new eyes, and a lot of times they can see things that no one else saw. So there are a number of examples of how people made mid-career changes. Actually, the Nobel laureate Walter Gilbert started out as a theoretical physicist, did some work that is fairly well known today related to the Higgs mechanism, just before Higgs worked on it. But he changed to molecular biology and won a Nobel in that field. So I think that's an example of how people can change and succeed in an area.
That's phenomenal. And at the time, you mentioned that you did your PhD at UCSD, and then you spent some time doing a joint postdoc at NIH and Johns Hopkins. What made you switch to go to industry?
Well, what happened there? Let me backtrack a little bit, because it was related to one of my two postdoctoral mentors, Solomon Snyder, and he got Warner Lambert Park Davis interested in adenosine. So the postdoc I did was actually John Daly set it up. John was someone who had already worked on adenosine for quite a while, and I was planning to come to his lab, and he set up a joint postdoc for various reasons, including financial, because I had gotten an extramural NIH fellowship, and it didn't pay as well as an intramural NIH fellowship. So to supplement that, he worked out a joint postdoc. So it was a lot of kind of figuring out how to get things done. But it was a very interesting combination, though, because John and I both knew a lot about adenosine. For Sol, it was his new toy, and he actually had some ideas that were very, very interesting. And in fact, when I have a Fitbit, and when I look up my sleep in the morning, a lot of times it gives me a little message that says that your sleep is caused by a molecule called adenosine, and that actually comes from some of my work in Sol's lab, and some experiments that Sol actually thought up. So the statement isn't completely true. There are other things that are important for sleep, and adenosine may be involved, but it's definitely, we're pretty sure now that at least when caffeine keeps you awake, that's due to blocking adenosine. So obviously adenosine is having its role in the whole thing.
Absolutely.
So
Yeah, please. We
Yes.
were
Okay.
backtracking
Good.
because I had an addition. Well, I think the question is going to come up later. But it's related to your favorite GPCR. But since adenosine receptors were the first ones that you read about and you got interested in and ended up working on, but I don't want to stop you. I want to let's
Okay.
continue in the
Yeah.
backtracking.
Hmm. Okay. So what happened in terms of my postdoc was that um, I'll just kind of mention as background, both John Daly and Sal Snyder were students of Julius Axelrod, who is a Nobel laureate. Uh, so in terms of scientific lineage, I'm actually Axelrod's double grandson, strangely enough. Um, and there's even a book called Apprentice to Genius, and it's about how Bernard Brodie, who was also a Nobelist, mentored Axelrod, and then Axelrod mentored Daly and Snyder, among quite a few others. And Snyder's students actually include a pretty long list of accomplished scientists, also. Uh, and I'm actually, I just wanted to mention here that I'm actually kind of disappointed that Sal never got a Nobel. I felt he deserved one, and it would have kind of added another generation to the whole story there. So that's just kind of a comment I'd like to throw out. So working for John and Sal, we did a couple of things. I developed the first adenosine receptor binding assay in 1980, although two other groups reported essentially the similar binding assays just a couple months later. So this is one of those cases when there was something in the air, and clearly a number of people kind of pursued this idea and came up with the results at about the same time. The other thing was that we made the case, as I just mentioned, that the stimulant effects of caffeine were due to blocking adenosine receptors. And this was kind of actually Sal's idea. John and I, having been working in adenosine for a while, knew about caffeine blocking adenosine receptors, and pretty much everybody in the adenosine field kind of assumed that was the case. But talking to Sal, he said, "Okay, but nobody's actually proven it." So Sal went and found somebody that can do some behavioral experiments, and we picked some analogs of caffeine, and we found that yes, the stimulant activity correlated with ability to block adenosine receptors. We had theobromine is a compound that's in cocoa. It's a close structural analog of caffeine, but it doesn't have stimulant activity. And so it seems like there's a correlation here with the adenosine receptor blockade. We also tested some adenosine analogs in mice as well, and they had really robust effects. The animals just completely stopped moving. This was an adenosine A1 receptor agonist, and so it got it. We didn't know if it would get in the brain, but obviously it did. The animals completely stopped moving, and they would sit there on their bellies with their eyes open, not moving at all. So it's really a very striking behavioral effect. So this was an interesting thing, and it's also one of my more cited papers. I guess I could bring up at this point. I kind of have some notes and thinking of things I want to say. So I could bring up at this point a saying by Louis Pasteur: "Chance favors a prepared mind." And later on, when we found by accident the adenosine A1 PAM, I knew what it was because of some experiences I had in the Snyder and Daly's lab. In particular, on the lab bench right down from me was someone named Moshe Gavish, who was working on GABA modulation of benzodiazepine receptor binding. This is an allosteric effect. Ken Murphy was working on binding to 3 different sites on the calcium channels that all interacted with each other. There's a verapamil site, a diltiazem site, and an nifedipine site. And those are 3 different sites, but they all have interactions with each other. And then from John Daly's side of the whole thing, John had a very interesting career. He was a chemist, and he would travel to Central and South America, um, finding these very colorful poison frogs, poison dart frogs, and he would extract the toxins from them and characterize their structures. That's actually how we got into adenosine research, because one of them was the trichotoxin, which is a very powerful opener of sodium channels. It causes convulsions, and you get big release of adenosine when you put the trichotoxin in with brain slices. So that was how John got into the whole adenosine field in the first place. But he had a collaborator, Edson X. Albuquerque, at University of Maryland, um, and who is an electrophysiologist. And there was another frog toxin called histrionicotoxin, and that was I believe, a NAM, a negative allosteric modulator at nicotinic receptors. So there were all these examples that I was exposed to during my postdoc of allosteric modulators of various types. So I kind of still had that in my mind later when I went to Warner Lambert, and we stumbled across the adenosine A1 PAM.
Oh, phenomenal. What made you, what made you switch from academia to industry? I think, especially, and correct me if I'm wrong, but for when I was doing my PhD about 10 to 15 years ago, going into industry was like going to the dark side. So I guess my question is twofold. One, what made you decide to switch from academia to industry? And at the time when you made the switch, was it considered as going to the dark side?
To some people, it was. It was interesting. I sometimes went out to ASPET meetings and so forth, and tried to recruit people for positions, and some people very obviously thought it was the dark side. And the thing is, if the world needs new drugs, who's going to do it? And unfortunately, government and academia are not so well suited for doing that sort of thing. And private industry has the resources, has the experience, and it can get these things done. So that's the upside of the whole thing. And there's also, of course, a downside as well, that you're working for a business. The business makes business decisions. We'll see a lot of that when I talk about the dopamine D1 receptor PAM. So you're kind of at the mercy of those. You have to bend with the winds, et cetera. So there are pluses and minuses both ways. But I got at Warner Lambert Park Davis in particular because Saul was a consultant there, and got them very interested in adenosine. And we had some interesting things we did with adenosine there. An adenosine A2 selective agonist had a profile that looked a lot like an antipsychotic drug. So Warner Lambert became very interested in that. It turned out the side effects were such that the drug couldn't be developed, but nevertheless, there are some very striking behavioral effects. The compound caused a decrease in locomotor activity, but didn't cause ataxia. So whereas a lot of sedatives will just, like a benzodiazepine, for instance, animals will just like fall over if you try to make them balance on a rod or do any number of other things that requires a minimum amount of coordination. Whereas the adenosine A2 agonist, the animals, their movement was a lot less, but they still were able to orient themselves to keep a balance if they needed to keep balance, et cetera. So it was a different profile, and one that was seen previously in antipsychotics. So there was a feeling that adenosine agonists might be antipsychotics. And I guess even today, maybe if somebody found an adenosine A2A PAM, it might be of interest to check it as an antipsychotic. So that's kind of the story with how I got to Warner Lambert. I did apply for some academic jobs. I didn't find any by the time I got a job offer from Warner Lambert, and it turned out the day after I accepted the Warner Lambert job, I got another call from another academic institution. I'd already accepted the job at Warner Lambert. So, that was the direction I went. There are many advantages and disadvantages with the whole thing in terms of you can get like really big resources for working on something if the company wants to put their resources behind it. But on the other hand, you have to do a lot of convincing. There's a lot of politics, et cetera, something I'm not particularly great at. So,
thank you for that. I was just going to mention that the way I see industry versus academia, it's the same coin. It's just two different sides that complement each other. And I think the great example is that you just mentioned that Warner Lambert got interested in adenosine because Saul was consulting with them. So industry is very much interested in the type of work that academics do because they do have the luxury of going explore topics, explore receptors, explore all sorts of things. But then when it comes, as you mentioned, when it comes to taking a target and getting a drug into the clinic, then the experience and the funding behind it is definitely in the private sector.
Yes, definitely. And the private sector really uses a lot of basic biological research all the time. I mean, they're constantly reading the literature, looking for new targets. All the genomics genome-wide, GWAS, whatever that stands for, when they get a correlation between a genomic change and something in the human population. That's a big thing for companies in terms of saying, okay, maybe we have a validated target here. So definitely academic and government research feeds into the pharmaceutical industry a lot. And that's kind of the way the system, I think, works best.
I think
although
so
we do basic research in industry as well.
Yeah, I think more and more often there is a portion or proportion of basic research that can be done in industry with the scope of potentially getting it into the pipeline and even publishing it. I think there's more and more companies that publish their research, which is really neat.
Yes. Although I see the trend as going kind of the other way, that back when I started, companies like to really show off the research a lot, and they supported basic research, and we're very proud of their publications. And I think at least in big pharma, it's not as prevalent as it used to be. Interestingly, my most cited paper actually came from when I was at Warner Lambert. We have a paper in 1986 in molecular pharmacology where we showed that there were two different adenosine A2 receptors, which we called A2A and A2B. The genes were eventually cloned, and the gene names are ADORA 2A and ADORA 2B. But that paper has 1,200 citations, I think, because people cite it whenever they mention, that they're working on a particular receptor, wow. One of those two receptors. So yeah, that was a big publication. And I should mention that molecular pharmacology is my favorite journal, and it will show up repeatedly in other things I published later on.
I was just going to say that's one of my favorite journals as well. So he read my mind on that front.
Yep.
So yes. You how much time did you spend at Warner Lambert before moving on to Eli Lilly and Company? And what type of, if you can obviously share with us, what kind of work were you doing there that was maybe different or you mentioned learning computational chemistry. I'm really interested in the type of work that you've performed in both of your positions at both companies.
Okay. At Warner Lambert, I was there for 7 years. I worked on adenosine, but as my lab got a little bigger, we worked on quite a few other things, prostaglandins, leukotrienes. We had the first leukotriene receptor binding assay, for instance. Once again, was something in the air, and I think two other groups came up with a very similar assays at practically the same time. What else? Glycine receptors, GABA uptake, quite a few different things that were going on. And I was one of the biochemical, in vitro guys. So a lot of that came through my lab. So a lot of different things. And it was actually kind of similar in some ways to what I did later as a computational chemist, where I supported all sorts of different projects, cancer, diabetes, many different therapeutic areas on different projects with all sorts of different mechanisms and different types of proteins they were interacting with, et cetera. So yeah, I did a lot of things. Let me see. Yeah, I guess I probably should go through this a little bit chronologically to kind of show how things went from one to the next. Yeah. So if it's okay with you, I'll talk about how we found the first GPCR PAM. And that happened, started, I was at, this was a couple years after I joined Warner Lambert. We were testing various A1, adenosine A1 antagonist series in 1983. And one of the compounds had a kind of a hump-shaped curve. It first went up by about 15%, and then it crashed. It went down a lot. And as a technical detail, this was a binding assay with an agonist radioligand. It would not have worked if it was an antagonist radioligand. So we were lucky we had a really first-class adenosine A1 agonist ligand that we're using there. So we might have ignored this result because it really wasn't much, up 15%, no big deal. But the assay was very reliable and the curve was smooth, despite the unusual shape. So we kind of kept it in mind. We were busy with other things at the time, but we went back to it a year later and we started looking at it. And from the experiences I already mentioned in the Snyder and Daly labs, I knew it could be an allosteric potentiator. So I was already kind of prepared for it. And what we interpreted the rising part of the curve as allosteric enhancement or positive allosteric modulation. And then we interpreted the plunging part of the curve as being just plain old orthosteric adenosine antagonism at the adenosine site. And the compound was from a known series of adenosine antagonists. So that was quite to be expected. So we thought we had, just by accident, two completely different activities. So the obvious idea is, can you separate these two activities? Is there some way you can just see the allosteric effect without seeing the antagonist effect? So we tried a number of tricks, and there are 5 or 6 tricks. I don't really have time enough to go through them all, so I'll just mention one. If we switch from rat A1 receptor rat brain to human brain membranes, the human brain has a much poorer affinity for this antagonist series. So now that unmasked a lot of the allosteric potentiation that was going on. And instead of getting a 15% increase in binding, now we got a 200% increase in binding. So it's like a quite robust effect now. So, and we had several other tricks similar to that. And they all showed us that we could look at this allosteric effect and in various ways mask out the antagonist effect. We never could get rid of it completely in the series we had, but we could go a long way in that direction. So things were looking interesting at this point. And very late in the whole thing, probably just like 3 or 4 months before I actually left for Lilly, we finally got a functional assay going, and we found a robust functional effect, about a 19-20-fold leftward shift in the concentration response curve to an adenosine agonist caused by this PAM. So then we knew it was for real. It wasn't just some artifact of binding. So we were very encouraged by that. So in interpreting our results, we used something called the Monod-Weinman-Changeau allosteric model from 1965. It's really the first publication on how allosterism worked. And the idea here is that the PAMs will bind selectively that the receptor or an enzyme in the case of the original studies
Yeah.
had two different conformations. One was inactive and one was active. And so the PAM would bind selectively to the active state. You can think of it as the active state. The protein may spontaneously switch between the inactive and the active state, and you can bind a PAM to the active state, and then it can't switch back. So it's stuck in the active state then. That's one way to think about it, and it's probably the easiest way to imagine it. There's actually a literature on what they call conformational selection versus conformational induction. And conformational selection is when, like what I said, the receptor spontaneously flips into the active state, and then you jam it in that state by binding something, agonist or PAM, either one. Conformational induction is you bind to even the inactive state, but then there's such a strong preference for the active state that it pushes the receptor over into the active state. And both of those could probably happen at equilibrium. It doesn't matter what the route is. It only matters what the final state is. I think in terms of practice, probably conformational induction is more important because it has faster kinetics for regulation. But that's a kind of an interesting story in its own right, and there's still a lot of debate about that. But for imagining how it works, conformational selection is, I think, a little easier to imagine. So that's how PAMs work, and in fact, that's also how agonists work. Agonists are actually allosteric modulators as well. We just call it the orthosteric site because it's the site that binds an endogenous agonist, and has been kind of fine-tuned over millions and millions of years of evolution. So they actually both work the same way. And free energies of this are additive, and what that means is the effects on affinity are multiplicative. So each one will potentiate the other. So you can think of a PAM as potentiating an agonist, but it's equally valid to think of it the other way around as the agonist potentiating the PAM. It's a very symmetrical situation. So we had all that in the paper we wrote back in 1990 in my favorite journal. We also speculated on some possible therapeutic applications of PAMs. And we thought this was very important. At low doses, they potentiate the endogenous agonist, but don't do a lot on their own. So what that means is they would only be active when and where endogenous agonist was present. In other words, when an agonist was released and where it was released. So they would actually amplify physiological control circuits. So their activity would be much more natural than with just giving an agonist, which would basically run around activating every receptor it can get to all the time. So we thought there was an advantage here in terms of fewer side effects because of that. And in addition, we would predict there would be a natural ceiling on activity, because if the organism gets overstimulated, it can stop releasing the neurotransmitter or hormone or whatever it is that we're potentiating there and get back to balance. And there could be also less desensitization because the effects, the agonist would be released in a transient manner, so the receptor wouldn't be activated all the time. Yeah. So we thought of all these advantages.
Fred, how novel?
Published 2 papers.
Sorry, please.
Mm-hmm. Yeah, okay. We published 2 papers in pharmacology in 1990, and they're not my most cited papers, but I think they're my most important papers.
And I think these arguments that you put forward are still valid today. I think we're still trying to figure out, can we use allosteric modulators? It's a very hot topic today. How novel were these ideas back in the 90s?
Fairly novel. There were such things known at ion channels. Benzodiazepines, for instance, are positive allosteric modulators of the GABA receptor. And interestingly, and also barbiturates are positive allosteric modulators at the GABA receptor. The difference between the two Barbiturates have two sites on the GABA receptor they bind to, and probably because of this, they actually have full agonist activity. They can activate the receptor without even having any GABA present, and that's why they're dangerous. That's why they're not used much today, and that benzodiazepines are more safe. There's only one benzodiazepine site, and the compound acts more like a potentiator, like a PAM, and is probably even a partial PAM. So, it's impossible for benzodiazepine to kill you by itself. Now, if you combine it with something else, like alcohol, for instance, then it can be dangerous. But at least by itself, it's much safer than barbiturates for exactly that reason, because it's a PAM, and just potentiates your endogenous GABA system. So these ideas were sort of known, but I don't think anybody really capitalized on the idea this is a way for the pharmaceutical industry to go to find something that's very selective and safer. So that's one thing I tried to put in the molecular pharmacology papers, how this could be a really nice thing in terms of how to work on different targets. And something else that's also an advantage here that I really didn't think of at the time, but that study was a good example, was that these sites usually are not so conserved compared to the orthosteric site. So what that means is a lot of times your compounds can be very selective for one receptor subtype versus another one. So for instance, we had a lot of trouble. We eventually succeeded, but we had a lot of trouble building A2 selective adenosine agonists, or by building any sort of selective adenosine antagonist. But right out of the bottle, our PAM was completely selective for the A1 receptor. And that's probably because that site is not so conserved, and the molecule was binding to some amino acids that weren't conserved in the other adenosine receptors. And that's true in a lot of cases. And the dopamine D1 PAM, it's practically completely selective for the D1, and doesn't interact pretty much at all with any of the other four dopamine receptors.
I love it. It's still contemporary. I just went last month to the GPCR's Targeted Drug Discovery Summit, and we had a bunch of talks regarding this topic, and the arguments that you just listed in favor of PAMs are still valid, and that's what I've heard as well in these talks. So how cool is this to hear that in the 1990s this was something that was already put forward, and we're still working on it.
Yes, it is cool. On the other hand, it's not cool that we haven't had a lot more drugs come out of it than we actually have. So that has been a disappointment. And then actually that kind of brings me to another topic I wanted to talk about, which was leading into what happened next in my career. And I was kind of expecting what I would almost call a triumphal march at this point. Now that we'd found a GPCR PAM, we had a proof of principle that such things exist. And therefore, you could pick a target and screen for a PAM, do some optimization in terms of the chemistry, get the compound into the clinic, and then go back and pick another target and do the same. And that was kind of how I thought things were going to go. And things unfortunately didn't go that way for my career. And that was really for three different reasons. And I'll get to, in this processes, I'll get to how I ended up being a computational chemist as well. So the first thing is Warner Lambert canceled the adenosine project, and I moved to Lilly. As I mentioned, Saul Snyder was a consultant with Warner Lambert, and when they dropped him as a consultant, they lost interest in adenosine. And this was actually the first of maybe a half dozen times when my career or my project took a nosedive, when a high-level sponsor or patron left for something else. So that's kind of a pattern that has repeated itself, unfortunately. In addition to that, the second thing was I joined Lilly to run a high-throughput screening lab, kind of similar to what I was doing at Warner Lambert. But that job disappeared when Lilly bought a small company to do all their screening. So I moved over to the central nervous system division, but I think they weren't so enthusiastic about me, given everything that happened later. And the third thing was more personal. My wife had some mental health issues, and the situation just basically became unlivable. So I ended up getting divorced. And for the next 5 years, I was a single dad raising 5 kids on my own. So it was a tough time. I had to throw a lot overboard during this period. And I actually, even today, cringe at some of the things I sort of left behind, things I never published, things I could have done. But it was unfortunate, but obviously that my kids were the first priority. And I guess the thing you can say is your job will never love you as much as your kids do. So, you have to pick your priorities. So that's what happened. And when things didn't go quite right, I did not have the bandwidth to do some of the things you would normally do to get straightened out and get in the direction I wanted to go, unfortunately. So anyway, my career got really weird at this point, and I ended up as a computational chemist. So I guess the interesting question here is, how did all that happen? When I got to Lilly, I started asking questions about what compounds my lab was screening. And it turned out there wasn't any plan. They just screened everything that the chemists had registered into the chemical database the previous week. So I taught myself, like I taught myself to program in Fortran, I taught myself to program in the chemical database language that was being used at Lilly. And I began selecting compound sets for screening. We called them cassettes. So my first cassette was related to non-peptide antagonists to neuropeptide receptors, which was a hot area at the time. So we successfully found leads for several neuropeptide receptors. These include the NK1 receptor for substance P, the NPY1 receptor for neuropeptide Y, and the V1A receptor for vasopressin. And the V1A series eventually made it into clinical trials. But that's really a long, complicated story that I don't have time to go into here. So I'll just leave that. There was a lot of kind of interesting stuff I did on neuropeptides at the time. And just as a side note, in terms of this period of my career, as I started working on selecting compounds, I coined the term virtual screening in a poster that I gave at a combinatorial chemistry meeting in 1995. I think that may be the first time anybody's used that term, virtual screening. So for what it's worth.
So anyone who uses the term virtual screening can send you a PayPal or a check for using that. Yeah, $1 for each time they would use it, because nowadays everybody wants to do virtual screening and in silico screening, because that's you can't. But physically you cannot screen billions and billions of compounds unless you do it virtually, basically.
Mm-hmm.
Yes. I wanted to ask, before you mentioned computational chemistry. You mentioned that the compounds that were screened were coming out of from the chemistry side that was input, and then everything was screened. And I understand your need to learn computational chemistry to be a little bit more specific and pick the right molecules, or at least look at the molecules that were screened and try and predict or select the ones that might give a desired activity. Where did you find the time to learn computational chemistry?
I just taught myself. I don't know how I did it. I just had I just started fiddling with the software, and the next thing you know, I was doing it. And over the years, I kept refining the methods and so forth. So for a long, long time, I was the person at Lilly who selected all the compounds for screening. It was a necessary thing to do because back when I started at Lilly, we had a couple 100,000 compounds in our collection, either made by our chemists or purchased from outside vendors. So that was more than we could screen in any assay. So we needed to cut it back fairly strongly, looking for, compounds that were desirable, compounds that might have a desired activity, and also looking to make sure that we had diverse compounds. We weren't screening two compounds that were practically identical to each other. So that's kind of all. Those are all the sorts of things I did back then. Was bring those methods to bear in terms of selecting the compounds. So in terms of my career, neuroscience management was uncomfortable with my dual role. So in 1997, they told me I needed to switch to computational chemistry full time. So from then on in my career at Lilly, I was at least formally a computational chemist. So I like to, at least keep my hand in terms of advice to different projects in terms of the biology side as well. So, for me, it was very sad because I had envisioned compound selection as a tool to support my receptor biology work, but the company didn't see it that way, and you have to kind of go with what the company wants. So I became a full-time computational chemist back then.
And how long were you a computational chemist before moving back to the lab side?
I never actually moved back to the lab side. I was still a computational chemist when I retired, but I was giving a lot of advice, in particular to the D1 PAM project. So I was really kind of working sort of almost on the side as an advisor to them on everything about the project, even though I was supposed to just be the computational chemist for the project. So I guess if this is a good time, I could launch into that, which is kind of the other bookend of my career. So in 2001, a colleague of mine named Charles Yang started a program looking for dopamine D1 receptor agonists to improve cognitive deficits in schizophrenia. There's already some literature about prefrontal cortex D1 receptors being potentially useful for improving cognition, cognitive abilities, possibly reducing things like delusions and other funny thought processes in schizophrenics. So Charles recruited me to the project to be the computational chemistry support. We first screened for looking for agonists, trying to find selective agonists that would be selective for the D1 over the D2. We didn't find anything that was selective enough for the chemist to be interested in. So with my urging, Charles decided in 2002 to go back and screen for D1 PAMs. So we had someone set up a D1 functional assay at our partner company that I mentioned already that we purchased several years before. And so it was a good assay in terms of throughput and so forth. And so we screened, this was very unusual at the time, we screened the entire Lilly collection, which was about 400,000 compounds by then. And we found one good hit out of the 400,000. Now, I mean, that's the hard part of the whole PAM thing is you're going in somewhere potentially completely new, trying to find something that you're hoping will exist, but you have no concrete evidence will exist. You're just looking for something. It's hard. We actually screened later on and we had better hit rates. So we did find other series, but we actually stuck with that first series because it was actually a pretty nice hit in terms of its potency and many of its properties. So we had some success in optimizing the series, but we also had big problems translating that into animal models. And the reason for that was that the rat and mouse D1 receptors had about 50-fold lower affinity for our PAM series than the human receptor. And it turns out that this is due to a mutation that occurred in the ancestor of all the rodents. So all rodents, including guinea pigs, have that mutation. All other mammalian species don't have the mutation. And in fact, Xenopus, Drosophila, they all don't have that mutation. So the human version is actually the ancestral version of that particular amino acid.
Wow.
But we didn't know that at the time, but we were struggling to get activity in animal models. That was really holding us back at that point. So anyway, in 2004, Lilly decided to get out of developing drugs for psychiatric indications, just a business decision. And the neuroscience division, yep.
No, I was just going to say that happens. And as you mentioned, that's something that you can't control for in spite of all the good data that you generated. And I find it so interesting that you screened 400,000 compounds and you got one hit.
Yes, that was, yeah, it was really amazing. Interestingly, we only screened 800 compounds to find that adenosine A1 PAM that we weren't even looking for. So it makes me wonder, was there something about the assay that was different? Were we very, very, very lucky? Or was there something about that particular assay? We did go up to much higher concentrations in that binding assay than we could ever go up to in a functional assay. So maybe that made the difference. I don't know. But that's definitely something to think about. But in any case, that is a big stumbling block for doing projects with PAMs, is that it's hard to find those initial hits. And sometimes they are difficult to optimize chemically as well. But I think it can be done. Certainly we did it with D1, as we'll see here. So going on here, so we lost the support of the project from the neuroscience division. So we changed the focus of the project to obesity, because D1 agonists were known to very strongly lower appetite. There's plenty of literature, for instance, amphetamines lower appetite quite strongly. So there's plenty of evidence that D1 agonists, the D1 receptor activation lowers appetite. So we did have a valid starting idea for this. So we basically, the whole project joined the endocrine division, and there was that was in charge of the obesity program. And an important development here was that endocrine authorized the creation of a transgenic mouse having the human D1 receptor. This is kind of an expensive thing to do at the time, and we did it with an outside contractor, and it took 2 to 3 years. So in the meantime, we had to live with whatever animal models we had. Another important development at the time was that John Schaus joined to lead the program. John was a high-ranking scientist on the scientific track in chemistry, and he was very good both at the scientific side of it and the political side of it, which is I think what we needed even more than the scientific side. So John was the one who kept the project alive and going for all those years from there until when we got a compound approved to go into the clinic. So his role is very important. Anyway, another one of these business changes. In 2007, then the endocrine leadership turned over, and the new leadership dropped obesity as an indication. It's kind of ironic, seeing how it's all the hullabaloo right now about new Lilly drug that has such strong appetite weight loss results, but at the time it was felt that this indication, obesity, was more of a cosmetic thing than a medical thing, and the company felt they needed to focus on diabetes. So they kind of got back into it actually through the back door through diabetes. But it's just kind of interesting historically that these things happen. And at that time, Charles Yang, the person who originally started the project, left Lilly and took a top job at ChemPartner, which is a contract research organization in Shanghai. And he'll eventually come back. and work with Lilly on monkey models for the D1 project. So even though he wasn't at Lilly, he once again started contributing to the project and did some very important work in terms of keeping this project going and reaching some success. So anyway, the project was terminated by endocrine, but the transgenic mice showed up in just barely the nick of time, literally weeks, wow, before it was too late. And there's someone in neuroscience, his name was Kjell Svensson. I'll spell his name out. He's Swedish. His first name is spelled Kjell. His last name Svennsson. So his lab worked on various neurochemical and behavioral types of models in animals. And what his lab found was that there were very robust effects of our compounds on locomotor activity in the transgenic mice with the human receptor, but not in the wild type mice. So that was why we'd had so much trouble all these years, was because we didn't have the human receptor in there. And if the mice had a receptor that was the same as the human, we could have had success many years before. But as it was, we took the kind of long and expensive route of developing a transgenic mouse. And that was really the key to the whole project, getting back on track. So neuroscience was interested in the project to work on cognitive deficits in Parkinson's disease. There was a big push toward neurodegenerative diseases, in particular Alzheimer's, but also Parkinson's as well. And our compounds, as we characterize them more, looked really good for various aspects of Parkinson's disease. Now, Parkinson's is not just a motor disorder. There's also a number of other impairments as well. There are cognitive impairments. There are mood impairments. There can be dementia and psychosis. So there are many other non-motor symptoms of Parkinson's. And our main focus was on the cognitive symptoms, but as you'll see, we also looked at other non-motor symptoms as well. So we made it to declare a clinical candidate in 2013. The generic name of the compound is mevidalin. It's very interesting. The generic names are arrived on through negotiations between the sponsoring drug company and an organization called the USAN. There are ideas back and forth about what to name it. I think the mevidalin actually comes from methyl, which is one of the groups on the molecule, VI for vigilance, D for dopamine, and then alin for allosteric enhancer. So that's how things get named, and presumably any other D1 PAM would be something dalin, because they usually keep the last part for the entire class of molecules. So that's how it got named. So going on here, I have quite a bit I could say about the D1 project. And let me just say our clinical biomarker was induction of wakefulness. In other words, we, in normal volunteers who'd been sleep deprived, the molecule helped them stay awake. So it had a quite robust effect on that. And that in itself could be a therapeutic indication as well, because there are vigilance-enhancing molecules, drugs on the market, and in fact, the attention deficit disorder molecules on the market all have this vigilance-enhancing effect. So that could be a therapeutic application right there. Anyway, the phase 2 trial was in Lewy body dementia patients, which are mostly Parkinson's patients. Unfortunately, it missed the primary endpoint, which was cognitive improvement. But it had, I'm reading from the actual papers published here, yep, dose-dependent positive effects on motor and non-motor symptoms, including alertness, daytime sleepiness, and possibly psychosis. So it looked like it had quite a number of other activities, including some that looked like there might be some promise for other indications that not related to Parkinson's. The interesting thing here is that cognition clinical trials are famous for their failure rate. Cancer trials, in terms of therapeutic indications, are well known to have the highest failure rate of any therapeutic area. But cognition clinical trials are even worse than cancer trials in terms of the success rate. So it was a high bar. We didn't make it over. So that was disappointing. And at this point, I don't know quite where things are. The phase 2 trial was completed in July 2020. On the Lilly website, the compound's still listed as being in phase 2. So I don't know quite for sure what's been going on there. I hope things are going forward in one way or the other.
It's almost
so in terms of my,
I was just going to say, it's almost a miracle that it went so far out, because this project on the D1 receptor was bounced around between all sorts of teams, changed indications so many times, but still, I think it shows, one, the persistence of the team that worked on this project, two, the fact that how cool are GPCRs, because you can basically not necessarily switch indications, but you can apply it to different indications. And I'm just glad that this went around and made it to some extent.
Yeah, I agree. And there are a number of, there are like articles written about the pharmaceutical industry, and there are a number of projects that were like this that got killed repeatedly, but the team really believed in it and kept bringing it back, and eventually it made it. And that's what happened here. The team itself was very much believing in this mechanism, and that our compounds had something to offer, and that was why we kept keeping it alive and bringing it back. And John Schaus and Shel Svensson were very critical in doing that. So the next part of the story is that how are we doing on time?
We're okay. We're at the top of the hour, but we're okay. Please go on. This is so interesting. I don't even want to stop you, because this is so interesting. I think you're one of our first guests who had this really concentrated experience in industry, but also navigating all these projects and all these receptors and all these molecules. And I will ask you later at the end of the story, what is your favorite GPCR? But there's so many that you worked on. So I think the answer, I can't wait to hear the answer, but please continue.
Okay, yeah. I've read your script, and I have an answer, but we'll get to that later. So I think the next thing in the D1 story is that I retired at the end of 2014. I signed a confidentiality agreement and set out to start writing up all the results from the D1 project. We wrote on a tool compound rather than the clinical candidate, because it wasn't the right time to start publishing on the clinical candidate. But the tool compound was practically identical to the clinical candidate, except not quite as potent. So basically anything we found for the tool compound, we also found for the clinical candidate. So the main purpose of all these publications from Lilly's point of view was to garner academic support, at least from the team's point of view, was to garner academic support. And hopefully people from outside the company would come in and say, hey, look at this, what you got. Isn't that something really exciting? Maybe we should look at schizophrenia, for instance. So we were hoping to get some academic buy-in to influence management to support the clinical development. So we ended up writing a number of papers. There are 4 that I'll talk about here that I was the primary author on. The first was a paper in Journal of Pharmacology and Experimental Therapeutics in 2017. This was related way back to that 1990 paper that talked about some potential advantages of PAMs. And I should mention here that the D1 agonists had problems in terms of being developed because they showed tachyphylaxis. Their activity waned when you dose them repeatedly. And they also showed inverted U-shaped concentration or dose response curves. In other words, at the high doses, the animals stopped. They had increased locomotor activity at intermediate doses and increases in other things of interest at intermediate doses. But at the high doses, that activity went away. And basically, as it turns out, that drop off was due to something called stereotypy, where if the animals get overstimulated, they start doing repetitive behaviors, in this case, repetitive grooming, and they stop, for instance, moving around the cage or doing the other things we were looking for. So our prediction was that our PAMs wouldn't show these effects because it would be related to the activity would be dependent on endogenous dopamine being released to D1 receptors. And that's what we showed in that paper, that we didn't see the tachyphylaxis and we didn't see the inverted U-shaped curves. The curves went up and didn't go back down. So this really confirmed after all these years the advantages that we predicted back in the 1990 paper. And surprisingly, after all these years, it's not that often that these theoretical advantages were actually confirmed in terms of real experiments in animals. So that was really important. In a second paper that went out in neuropharmacology in 2018, we focused on therapeutic indications for D1 PAMs. We were using the humanized D1 mice. We also did some work in rhesus monkeys that Charles Yang was involved with, and we showed in the transgenic mice that our compound was active in a number of models that predict utility in indications such as schizophrenia, depression, and vigilance enhancement or attention deficit disorder. So what that tells us is we have a lot of scope for different ways to go with the D1 PAM. So that's important in terms of possibly providing inspiration for the clinical development. Okay, a third paper I'll talk about in a minute. It was in my favorite journal, but I want to go back a second to 2004. In 2004, we did some rat-human chimera constructs, and we found that the compounds bound to intracellular loop 2 of the D1 receptor. And this intracellular site at the time, as far as I know, was the first time that a PAM site had been reported for an intracellular site. So it was a big surprise to us. But unfortunately, the compounds hadn't been patented, so there's no way we could publish. If we published, we would have blown the ability of the company to patent the compounds, and therefore the compound series could never have been developed. So we had to sit on it, and we sat on it for, it turns out, 11 years. Well, actually, for us, it was, more than 11, 14 years. In 2015, anyway, a group from Bristol Myers Squibb scooped us on this particular thing because they had a D1 project and they showed one of their D1 PABs bound to the site. So we lost our novelty there. But we ended up publishing our own data finally in 2018 in molecular pharmacology. So we showed, interestingly, we wanted to have some sort of we did a number of mutagenesis experiments and built a homology model. And we wanted to have a control compound, and we found something in the NIH database that David Sibley's group had done. They were screening for D1 PAMs, and they had some compounds listed in there from their primary screening data. And we went back and looked for analogs in the Lilly collection and found a compound that looked even better than what they'd reported. And unbeknownst to us at the time, Sibley's group had done the same thing and found the same compound. So about a year before we published, we contacted Sibley's group and said, hey, we're planning to publish on your compound. So just to let you know. First of all, tell us, when you're planning to publish, and, hopefully we can kind of, at least know that each of us is working on the same thing.
Yeah.
It would have been very rude for us to just scoop them on their own compound. anyway, what happened was we published in Molecular Pharmacology, and in parallel, Sibley's group had been working on their compound, and it turns out they also had an ICL2 binder. So we both published in the same issue of Molecular Pharmacology, somewhat unknown to each other, because we even went through different editors, and the editors of the journal didn't know either until the last second when we saw the preprint coming out from Sibley's group, and ours was in proofreading at this point, in page proof. So we hurried it up and got ours into the same issue. So another example of like the same wind was blowing. But it was interesting. In both cases, we found that the two PAMs actually multiplied each other in terms of their effects. So you could get a much larger shift in the dopamine curve with both of them combined than with either one of them. Or in our case, we showed that the two different PAMs, because they bound to different sites, had a synergistic effect. And even though either one of them separately had very small effect on its own without dopamine present, together they had a much larger effect. In our case, it was 2% effect plus a 2% effect added up to a 23% effect. So there's a big synergistic effect there. So that was really interesting. And finally, the latest paper, which is going to be, I think, probably my last paper, just came out in Molecular Pharmacology in the March issue. And that is showing that there's a third PAM site that's occupied by yet another PAM. And that PAM was actually another PAM that was in the 2015 Bristol-Myers Squibb paper. They'd actually reported two compounds, one of which bound to ICL2, and the other one which bound to an unknown site that wasn't ICL2. We took a look at that structure, and it looked very different from the Sibley group compounds. So we thought that maybe it was binding to a totally different site. Our chemists eventually were successful in making the compound, and after some delay, we actually got the experiments done. And indeed, it did bind to a different site. We had D1, D5 chimeras and showed that it bound to somewhere in the the extracellular N-terminal part of the D1 receptor, which was different from either of the other two PAMs. So the interesting thing was that, as you'd predict, any pairwise combination showed a multiplicative effect. So any of the three pairwise combinations, we saw that. And then if we put all three in, we got this humongous 1,000-fold leftward shift in the concentration response curve for dopamine. So that was a big deal because it kind of confirmed a lot of the predictions we made, that come out of the original Monod, Wyman, Changeau model of allostery. Yeah. So I think in my mind, the main point of the paper was to circle back to that Monod model that I'd used in 1990 and show that now we have a greater validation and try to get a better recognition of this model in the GPCR community. And in particular, a point that it made like pretty emphatically here was that any site on the receptor that changes its conformation, changes its configuration between the active and inactive state can be a host site for a PAM or a NAM, because a compound can have a different affinity for one conformation than the other. So this gives us a lot of scope to find different binding sites that a PAM or a NAM might work on. So I thought that was kind of a nice kind of bookend to my career. So I guess that's kind of it for my career. I do have two ideas I'd like to run by listeners if I have time.
Of course,
do I have enough time?
Yes, please.
Okay. There are two ideas I've been thinking about, and for one reason or another, haven't been able to pursue. The first one relates to the D2 receptor. I should mention here that there are 5 dopamine receptors. D1 and D2 are the main dopamine receptors. They're expressed at 10 or 20 times higher levels in brain than D3, D4, and D5. So the biggies are D1 and D2. And the D2 receptor is the main dopamine autoreceptor. So dopamine inhibits its own release through the D2 receptor. So blocking the D2 receptor causes a big increase in dopamine release, which then would selectively increase D1 tone because the D2 receptor is blocked and isn't available. So the question is, does this increase in D1 tone contribute to the antipsychotic effects of D2 antagonists? I included this idea in the neuropharmacology paper. And my question kind of is, how much is this understood or believed in the wider dopamine in schizophrenia community? There's a lot of literature showing increases in dopamine release, for instance, in the prefrontal cortex caused by antipsychotics. But the only paper I could find connecting that D1 stimulation to the beneficial effects in schizophrenia is something that was actually published by my colleague Charles Yang in 2001 in Journal of Neurophysiology. Mm-hmm. So I don't know if this is an idea that everybody kind of believes, but nobody's really nobody's really stated outright, or whether it's somewhat a novel idea. The literature is really complicated in this area. I'm not an expert. So, and I think probably the only way to sort it out would be a clinical trial with the D1 PAM. So I guess if there are any listeners that are interested in this idea, I mean, contact me or contact somebody else who would be interested in the idea or interested in doing something to test the idea, etc. So that's idea number one. Idea number two is related to the D5 receptor. The D1 and D5 receptors are closely related to each other. They both stimulate adenylate cyclase, rather the D2, D3, and D4 are linked to the inhibitory G proteins. And the D5 receptor is expressed at much lower levels, but it's conserved, so it must have some important role doing something. But that's not very well understood. So the interesting thing here is that there's a single amino acid that's responsible for the fact that our PAM has about 1,000-fold higher affinity for the D1 than the D5. So the idea here would be to make a double transgenic mouse that flips that particular amino acid. So you make the PAM inactive at D1 in this transgenic mouse. At the same time, you make it active at D5. And then you have a very specific tool to look at what the role of D5 is. So I guess if anybody's interested in this idea or the other idea I mentioned, please contact me or just pursue the idea on your own, whatever works. So
I'm happy to make introductions to a couple of scientists in the dopamine field. Last month, we dedicated our 4 episodes of the month to celebrating Marc Caron's legacy and scientific life. And I got to talk to a lot of people who are still working on dopamine receptors, have many mouse models. So I'll be able to make that connection. Who knows? Maybe these two ideas could be 2 great PhD or postdoc projects, which could be really interesting.
They could. I'd like to see what could come out of these ideas anyway. They've just been ideas that have been nagging at me for many years now, and I think this is a good forum to kind of get them out there and see if anybody can take them on. So I'm glad to be on this for that reason.
That's amazing. So, I'm happy to make the introductions, and anyone listening to the episode, if they feel like they would like to reach out to you, they can find your information on the podcast page once the podcast comes out. All right, 2 last questions, because I know we've talked a lot, and I didn't want to interrupt you because I think your career is amazing. You've done so many things, worked on so many receptors, but here it is. What is your favorite GPCR?
Okay, my favorite GPCR, actually my favorite molecule, you ask sometimes what for your favorite molecule. My favorite molecule is adenosine still, because it does so many things. It's part of RNA, it's part of ATP, it's part of NAD, and ATP itself is a neurotransmitter as well. So it has a lot of different things it's a part of. By the way, in terms of the role of adenosine, it's involved in energy supply demand balance. So if a cell starts running down its ATP from too much energy demand, some of that gets converted into AMP, which gets broken down into adenosine. It gets released, and the surrounding tissue senses it in terms of adenosine receptors. And then it lowers energy demand, mostly through A1 receptors, and it increases energy supply, mostly through A2A receptors, which are involved in vasodilation. So that, in a nutshell, is what adenosine is all about. But going on to my favorite receptor is actually D1. And the reason for that is I got to do with my own little fingers tapping on my own keyboard of my own workstation a homology model of D1. And it was based on an agonist state crystal structure of the beta-2 adrenergic receptor by Brian Kobilka's group in 2011. We were lucky that came up at that time, and we built a homology model of D1. I should mention that D1 and D5 are actually more closely related to the beta receptors than they are to D2, D3, and D4. D2 and D3 and D4 are more closely related to the alpha adrenergic receptors. So the beta-2 is actually a pretty close relative of the D1. So we built a homology model, and we docked our molecule in there. And I was happy to see in 2021 that there actually was a cryo-EM structure of the Lilly PAM series bound to the D1 receptor. So I actually got to see how it bound. And we nearly got it right. We had the correct conformation of the ligand. We had the correct binding mode or binding configuration, how it fit into the binding site. We had that pretty much correct, how it lay down in the binding site. The one thing that we missed was we had it about 2 angstroms further up toward the extracellular side than where it actually was. And the reason for that was there was an important tryptophan that was involved in a charged a stacking interaction with the group on the ligand, and that tryptophan was rotated by 180 degrees in the protein compared to how we had modeled it in. So the whole thing moved down by a couple angstroms to maintain this interaction with the tryptophan. But that was a great thing to look at. So that was why I think the D1 is still my favorite receptor.
How amazing is that? I think it's really cool. You can say, we missed it by 2 angstrom. We were a little bit off. we got the rest right without the structure, which I think is phenomenal.
Yeah, it was lucky we had a really nice structure of the beta-2 to work from. So yeah, if it had been some other receptor that was more distant, we couldn't have done as well. Yeah. But yeah, that was a good thing.
Last question. Any additional advice for young scientists aspiring to contribute to the field?
Okay, sure. I wrote some things down here. First thing, that you hear a lot about follow your passion, and I completely agree with that, but also think about following your skills as well as your passion. And I kind of wonder, even today, if I might have done better sticking to theoretical physics, where my math skills would have been really important. Math isn't your first thing for neuroscience in general, although I guess there are theoretical neuroscientists these days. And I think computational chemistry was kind of a halfway move back in that more mathy direction, as it were. And interestingly, a kid in my high school calculus class ended up being chairman of astrophysics at University of Colorado. So it really makes me wonder. Okay, that's one thing. A second thing is just keep your eyes open. Important discoveries often come from serendipity. I mean, you may have a research program, but you can't always force things to work the way you think they're going to. And sometimes a door will open that you didn't even know existed. So that was what happened with our first adenosine PAM. So, these things happen all the time. And then I guess industry versus academia, I think we've already heard a lot about it, but industry provides better resources for developing drugs, but your progress can be impacted by business decisions. And it can be harder to publish your work if you're in industry. Certainly, you may have to wait a long time. And sometimes you need a whole lot of patience. We started the D1 project in 2001. It's now 2023. So what, 22, almost 22 years later, and the compound still has not made it all the way through clinical development. So it takes an ungodly amount of time to do things, to get things done in industry. It's very highly regulated, and you need to be very careful as you move forward, especially in the clinical work, because these are real people that you're testing your drug on.
Yeah.
So, and I already mentioned I had some advice to mid-career scientists about trying new things, and old brains with looking at new things can get new ideas. So that kind of happened to me with the computational chemistry, and I think that's something also that's quite important as well. So I guess
I think
that's
too.
what I have to say.
I think so too. I think that's amazing. Fred, thank you so much for your time. I really enjoyed learning about all the projects you worked on and about how you were able to navigate all these programs across two companies in all these years. Thank you so much.
Yep, thank you for having me. It's been a great thing to be on.
Thank you. I'll talk to you soon. Thank you for joining us for this Dr. GPCR podcast episode. We would like to take a moment and thank our guests, as well as our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Please also don't forget to subscribe to the Dr. GPCR monthly newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. And until next time, stay safe.